Welcome back to section 17.4. We're going to look at the hormones and their actions. Here is the list of expected learning outcomes for 17.4. So we have three classes of steroids, of hormones. We've got the steroid hormones, the monoamines, and the peptides. The steroids are all derived from the steroid cholesterol. Our sex hormones, such as estrogen and testosterone, come from the gonads. We have our corticosteroids like cortisol that come from the adrenal glands. Our monoamines are made from amino acids, and these are our catecholamines like dopamine, epinephrine, and norepinephrine. Some of our other monoamines are melatonin, as well as thyroid hormone. So if we look here at testosterone and estradiol, we see that they have the same basic structure with these 4 carbon rings here. And what changes is whatever is hanging off of those carbon rings. If we look at the difference between testosterone and estradiol, we have a hydroxyl group here where we have a double bonded oxygen here. We have a methyl group here where we have no methyl group here. We have a set of double bonds here and we only have 1 double bond there. Otherwise, it's pretty much the same. Our monoamines come from a group of amino acids, and those amino acids have their particular amino groups, and then from there they have their side chains that are going to be different. Our peptides and glycoproteins are going to be created from chains of amino acids. These hormones are going to come from both lobes of the pituitary gland. We've got our releasing and our inhibiting hormones from the hypothalamus. Insulin is also a large peptide hormone. If we look at oxytocin, oxytocin comes from the posterior pituitary. It's a set of amino acids that forms this very peculiar peptide chain. Here is insulin, which is a very large protein that essentially is 2 polypeptide chains bonded together. So when we have hormone synthesis for our steroids, all of them are going to come from cholesterol. That cholesterol is going to be converted to progesterone. And from progesterone, we can then make our cortisol or our aldosterone. We can also make our sex hormones, testosterone and estradiol. But they all come from cholesterol. And what changes is going to be what we attach to or take away from that cholesterol. Our peptides are going to be synthesized the same way as any protein. They're going to be a gene that is transcribed off of the DNA to a messenger RNA. That messenger RNA is going to be taken to the ribosome where we're going to assemble a peptide from the amino acids. The rough endoplasmic reticulum and the Golgi body will modify these peptides to produce the mature hormone. Proinsulin is the inactive form of insulin. It has some connecting peptides that have to be removed in order for it to become insulin. If we look here, we've got our 2 structures of insulin here, and then we have this connecting peptide. And in the Golgi body, this connecting peptide will be snipped away and will release then the active insulin. The monoamines are going to be synthesized from amino acids. Many of them are going to come from the amino acid tyrosine, whereas melatonin is going to be synthesized from the amino acid tryptophan. The thyroid hormone is also composed by tyrosine. We have our follicular cells are going to absorb the ion of iodine from the blood, and it's going to oxidize it to a reactive form. The cells then are going to synthesize a large protein called the thyroglobulin, and they're going to store it there in the follicle. When iodine comes in, it's going to have the tyrosines are going to the iodine is going to be added to the tyrosines, and we're also going to add that thyroglobulin. When the 2 tyrosines are going to link together, we're going to have the forerunners of T3 and T4, the thyroid hormones. When the follicle receives thyroid stimulating hormone, it's going to have an effect on that thyroglobulin, and it's going to cause lysosomal enzymes to split that thyroglobulin off and free the thyroid hormone. Most of these thyroid hormones are going to come as T4. and they're going to be released from the follicle into the blood capillary. So we have our iodine coming in, and we're going to have the thyroglobulins that are going to attach to those iodines, and then we're going to have the lysosomal enzymes come and cleave that to give us our T3 and our T4, which will then be released into the bloodstream. So if we look at T3, we have 1, 2, 3 iodines, where here at T4, we have 1, 2, 3, 4. Here are the precursor tyrosines. We're going to bind those together through dehydration synthesis and get our T3 and our T4 hormones. Hormones are not going to be secreted at steady rates. Levels in the bloodstream are going to fluctuate throughout the day. Some of these are going to be secreted in cases on a daily circadian rhythm, some of them in a monthly cycle, like our FSH, our LH. Others are going to be under the influence of specific stimuli that are going to only be activated when we need them. We have 3 different types of stimuli. We've got neural stimuli, we've got hormonal stimuli, and we've got humoral stimuli. So if we look at neural stimuli, we see that the nerve fibers are going to supply some of the endocrine glands with their specific hormones. These sympathetic neurons are going to stimulate the adrenal gland to secrete epinephrine and norepinephrine in times of stress. In childbirth, we're going to see some of the nerves in the uterus are going to stretch, and they're going to send a message up the spinal cord to the brainstem, to the hypothalamus, which is going to stimulate the release of oxytocin from the posterior pituitary. Hormonal stimuli, we're going to have hormones from the hypothalamus regulate the secretion of the anterior pituitary. The pituitary hormones then are going to stimulate other endocrine glands to release their particular hormones, like the thyroid hormone, our sex hormone, and the cortex of the adrenal gland to release cortisol. Humoral stimuli is going to refer to a bloodborne type of stimuli. We're going to have raising blood glucose concentrations, which are going to stimulate the release of insulin, or low blood osmolarity, which is going to secrete the release of aldosterone, or low blood calcium, which will stimulate the release of parathyroid hormone. So we have a number of things that are going to be carried in the blood, or changes in the blood, that are going to initiate the release of hormones. Peptide hormones and catecholamines are going to be stored in specific vesicles that we call secretory vesicles of the particular endocrine cells, and they're going to stay there until we need them, and they're going to be released through a process of exocytosis. Our steroid hormones are not going to be stored in vesicles or released by exocytosis. They're going to be released as fast as they are stimulated, and they can be diffused through the cell membrane. Once they're synthesized, they can then act on the cell. The stimuli, such as follicle stimulating hormone or adrenocorticotropic hormone, can increase the synthesis of particular steroid hormones by 7-fold within a very short period of time. Thyroid hormone also diffuses freely across the plasma membrane. It does accumulate in the gland where it waits some kind of stimulus for secretion. It's not going to be stored in endocrine cells, but in extracellular spaces enclosed by those thyroid follicles. Once thyroid stimulating hormone is released, it's going to cause the release of these thyroid hormones from the thyroid follicles. Most of the monoamines and peptides are what we call hydrophilic. These are going to mix easily with blood plasma. The steroids and thyroid hormones are hydrophobic. These are going to bind to transport proteins, mostly albumins and globulins, that are going to allow them to have a longer half-life so they don't get broken down by enzymes of the liver or filtered out through the kidneys. The unbound hormones are going to leave through capillaries and reach their target. Once these transport proteins are going to protect these circulating hormones from being broken down, and then we will be able to use them later on in the bloodstream. Thyroid hormone is going to bind to 3 transport proteins in the plasma, either albumin, thyretin, or thyroxine binding globulin. More than 99% of our circulating thyroid hormone is bound to protein. Some of our steroid hormones are bound to globulins. Transcortin is going to be a transport protein to cortisol. Aldosterone has a very short half-life because about 85% of it is unbound. 15% of it is weakly bound to albumin. And this is going to give us that ability to control water balance and electrolyte balance so that we don't have issues of disastrous changes in blood pressure. We do have some hormones that stimulate only those cells that have receptors for them. Hormones all have specific receptors for their specific hormones, and those receptors are going to either be proteins or glycoproteins. They're going to be on the plasma membrane. They're going to either be in the cytoplasm or in the nucleus. These receptors are going to be switches that are going to turn on very specific pathways once hormones bind to them. Usually each target cell has a few thousand receptors for a given hormone. The receptor hormone interactions are going to be very specific and can produce something called saturation. The specific receptor to a specific hormone only allows that hormone to have any activity on that cell if it has a receptor for that hormone. Saturation is when all of the receptors are occupied by the hormone molecules themselves. We do have some peptides and catecholamines. These can't penetrate the cell. They're going to bind to some surface receptor and activate some kind of second messenger system inside the cell. The steroids and the thyroid hormones can penetrate the plasma membrane because they are hydrophobic. They influence the expression of genes that they target. This is going to then initiate some changes in metabolism in that cell. This can take several hours to several days to show any kind of effect due to the lag of protein synthesis. If we look at our peptide hormone here, we're seeing that it binds to a receptor. It's going to activate a membrane enzyme. That membrane enzyme is going to activate a second messenger, and that second messenger then is going to have some kind of metabolic effect. The steroid hormone is going to go straight through the plasma membrane. It's going to bind to a receptor on the nucleus. It's going to activate some kind of gene transcription, which leads to protein synthesis, which will then have some kind of varied metabolic effect on that cell. Our peptides and catecholamines are going to bind a very specific second messenger. These hormones are going to bind to a receptor, which is going to activate a particular protein called the G protein. That G protein is going to activate some enzyme in the membrane called adenylate cyclase, which activates our second messenger, cyclic AMP. That cyclic AMP is going to activate inactive kinases. Those active kinases are going to activate inactive enzymes. Those activated enzymes now are going to bind to substrates to produce products that give me some kind of metabolic effect to my cell. This is essentially the same. It's just showing you specific hormones and their activation of second messengers. Estrogen binds to nuclear receptors in the cells of the uterus. It can activate a particular gene for the progesterone receptor. Once it has that receptor, now progesterone can come in later during the menstrual cycle. It can bind to those receptors and stimulate the gene for some kind of nutrient synthesizing enzyme. Thyroid hormone is going to enter into the cell by means of an ATP-dependent transport protein. Once it gets into the cell, T4 is going to be converted to T3, which is a more potent thyroid hormone. T3 binds to the nuclear receptors. It activates a gene in the sodium-potassium pump so that we can now produce that sodium-potassium pump for our metabolism. Most of our cells are sensitive to more than one hormone and exhibit certain interactive effects. We can have synergistic effects, which are going to have multiple hormones acting together for a greater effect. An example of this is going to be follicle stimulating hormone and testosterone on sperm production. We also have some permissive effects where one hormone enhances a target organ's response to a second hormone later. Estrogen is going to prepare the uterus for the action of progesterone. Some also have antagonistic effects where one hormone opposes the action of another. Insulin lowers blood glucose where glycogen raises blood glucose. So if we look at the antagonistic effects, we have the absorption of our blood sugar. That glucose is going to go into the cell. The liver is going to cause the production of glucose through gluconeogenesis and glycogenolysis. That glucose then is going to be released from the cell, and we're going to have the elevated blood glucose. So the glycogen now is going to increase our blood glucose, which will then in turn cause the action of insulin, and we just have this constant lowering and raising of our blood sugar through the action of insulin and glucagon. We do have to clear our hormones, and most of them are going to be cleared through the liver or through the kidneys. They're going to be signaled to turn them off when they have served whatever purpose they were produced for. Most of our hormones are going to be, again, degraded by the liver or excreted by the kidneys. Most of them are going to be excreted in the bile, which will end up in our feces or in our urine. We have what is called a metabolic clearance rate, and this is the rate of hormone removal from the blood. All of our hormones have a half-life, and this is a half-life that is required, the time required to clear 50% of the hormones from the blood. The greater or the faster the metabolic factor, the clearance factor, the shorter the half-life. Okay, that's the end of 17.5. The last section of chapter 17 is 17.6. We're going to be looking at the eicosanoids and other signaling molecules.